everybody. Thank you so much for joining us today. We are so excited to get into neuromuscular reflexes. As folks get kind of settled in, I'd love to direct you to the chat box. We'll go ahead and use that to be asking questions today. I'm going to keep an eye on the chat, and Melville's going to keep an eye on the chat. So if you have questions, please feel free to pop it in there. I just ask that you set that to everyone. So go ahead and navigate to the bottom of your Zoom window and locate your chat bubble. It looks like a little, like a little chat bubble at the bottom. And if you click that, you can set that blue button to everyone. And that way everyone can see your questions. So Melville and I don't look like we're talking to ourselves when we answer your questions. As you're getting settled in, let's go ahead and break the ice. I was just saying that I'm going to a Lord of the Rings trivia night tonight, and I'd love to hear from you all. What is the movie, book, show, subject, etc., that you could go to a trivia night about and feel like you were going to get it all? So put that in the chat box and remember to set it to everyone.
And I was telling Dylan that a Lord of the Rings event would be my favorite to actually go to. So
That's right. All right, as folks are typing, I'll run through some quick housekeeping items and then I have a poll for you all, so I'll get that started. First and foremost, you will receive a recording of today's webinar. It usually takes us about 3 to 5 business days to get that edited, but as soon as it's edited, it will be posted on both our YouTube channel and our website. As soon as it's live, I'm also going to email it to you along with a resources folder that I'll drop in the chat right now. In that resources folder is the presentation that you're looking at right now with clickable links, as well as a certificate of attendance and some other goodies. So make sure you check that out. And the second thing that I wanted to mention to you all today is, as I said, we want to answer your questions. So if at any point during the presentation you have a question, want to run something by us, please feel free to put that in the chat. I'll do my best to answer it, or I will bubble it on up to the professional, and we'll get you an answer. If we happen to miss your question today, please feel free to reach out. You can email us at biology@vernier.com and somebody will get back to you. And it looks like Jack in the chat would also go to a Lord of the Rings event. So it sounds like we have our trivia team right here. And without further ado, I'll pass you off to our presenter and thank you again for joining us.
great. Thank you, Dylan. All right. So my name is John Melville, and what I'm going to do is just briefly go through the agenda, talk about a few things. And then what's great is if you have any questions or anything like that, feel free to ask them in chat. I can see the chat. So don't worry, just type it in chat and ask me a question. So just once again, questions are welcome at any time. The other thing that I wanted to let you know is that you can get access to this presentation and it has some links to online resources that may be useful for later. The other thing that would be great is if you could tell me, like, are you a K through 12 teacher? Just write that in chat. Oh, there's the pre-webinar question. So go ahead and answer the survey questions that Dylan is about to pop up, and because that'll be really helpful for when moving forward with where I focus this presentation. And then I just want to let you know that what I'm going to do is I'm going to explain and talk about our Go Direct sensors. I'll give you an introduction to graphical analysis, which is what most people would be using to collect data with now. And then I'll give a brief introduction to our human physiology books. And then I'll talk about a very specific experiment, which is our reflex experiment, which is very popular. Cool. Great. Okay, so that's what I thought. People are here to learn a little bit about the sensors, but most of you are here to learn about the experiment. Awesome. Thanks. That helps a lot. I really appreciate that. So just a little bit about me. My name is, like I said, John Melville. I've got a bachelor's degree in biology and psychology from Sonoma State University. I got my PhD in zoology at Oregon State University with a background in neuroscience. And then I did a postdoc at the University of Oregon, also in neuroscience. But I was also, more importantly, a teacher. So I was an adjunct instructor at Lane Community College in Oregon. And then I moved out to Iowa, where I was an assistant professor at Wartburg College, where I started several neuroscience courses, but I also taught majors and non-majors courses in biology and neuroscience. And almost all of my activities and my focus was on getting students to do experiments, do science themselves, and to come up with researchable questions. So I'm really big into inquiry. But one of the things that I did do is I started um, a neuroscience program, and that's really how one of the ways where I really got involved in Vernier, because my students really liked doing a series of experiments from our human physiology book, and we've modified that experiment to use with our Go Direct sensors. So one of the things that you might want to do right now, you don't have to, but I wanted to let you know, I'm going to be showing you graphical analysis. Graphical analysis is free. You can download it on your device. So if you go to our website, if you go to the Vernier website and just go to downloads, you can get a hold of graphical analysis. Um, graphical analysis can also run in a web browser. What that means is that if I were to take, oh, hold on here, let me go back, is that if you were to type this, http://graphicalanalysis.app in a Chrome web browser, graphical analysis will launch. So you don't have to download anything as long as you have a Chrome web browser, or you can get the actual download and download it to your Mac or PC. If you've got a Chromebook, you're going to have to use this. It also runs on iOS and Android, and you can get graphical analysis in your appropriate app store. Just make sure it's the latest version, update it if necessary, if you already have one, and don't worry, you don't need a sensor. We just might play around with some data later if you want to. All right, so if you launch graphical analysis, one of the things that you might want to do is we have a basic version of graphical analysis, which works great for just basic data collection, but I'm going to be showing you some actually pretty cool features that are in our pro version. This over here, where it says GA Pro key in the bottom, you can type this number in. When you launch graphical analysis, you'll see it says activate additional features. This little license key will give you GA Pro until the first week of November. So if you want to play around with GA Pro, you can do that. GA Pro is a subscription service, or you might think of it as a site license, but you can buy 1, 3, 5 years worth of that site license. And in some cases, it'll be really worth it to you. In other cases, it might not. For this experiment, it's actually pretty neat. There's a very cool feature that I'll show you that I think makes it worth it. All right. So first off, what I want to talk about is our Physiology Go Direct standard package, or our Physiology Go Direct sensors. So what you can see up here on the screen here is these are our Go Direct sensors. All of these sensors do not require an interface. So you don't have to use a LabQuest. You don't have to use a LabQuest Mini. They have a USB port and a Bluetooth radio. And we have a whole series of sensors that you can use. Blood pressure, a hand dynamometer, EKG sensor, there's a spirometer, oxygen gas, respiration belt. This over here is actually a force sensor that's attached to a reflex hammer, and I'll show you that in a minute. The other big thing to be aware of is, if you're going to be getting our human physiology sensors, is we have 2 human physiology books that are designed to be used with these sensors. We have Human Physiology Experiments Volume 1, which uses a subset, and that's where the reflex lab that I'm going to show you is. It's located in that book. And then we also have Human Physiology Experiments Volume 2, which is a much more advanced set of experiments that uses our blood pressure and spirometer, but also has some more advanced labs in it as well. So if you're getting the entire standard package with the spirometer and everything, I recommend getting both books. If you're just going to get a subset and you're not going to get the blood pressure sensor or the spirometer, then just get the first book. By the way, if you get a book, you only need to buy 1 copy for your school. It's basically a site license for you and any other teachers at your school if you get it. So that's another big thing about our books, is we're not expecting you to buy multiple, and it's really fine if you take the instructions, modify them, give them to your students, etc. So it's a really, really broad site license for the books. All right, so what I want to do next is just show you a little bit about the Go Direct sensors, how they work. How many of you have actually used a Go Direct sensor before? Just type in the chat yes or no. I just want to see how many of you have seen how they work. If you don't mind doing that real quick for me. Yes. Okay. Jack has yes. Peggy yes. Great. Okay, Eric, no. Okay, so we've got one person so far. No, everyone else, yes. Okay, so what I'll do then is I'll just for Aaron then I'll briefly go through how they work, but I won't spend a ton of time on it. But Aaron, we can always come back and I can show you more about how they work. That sound good? Great. So here what I'm going to do is I'm going to bring over this little camera, hopefully that you can see. And what I just want to show you is the Go Direct sensors, they do not require an interface. They all have a USB port on them. So this is a hand dynamometer. And so you can either just plug it in directly over the USB port, or they can be used wirelessly. So if I tap here on this little yellow button, it'll begin to flash, and then I can connect to it wirelessly. And let me just do a brief demo of that, and then I promise we will move on. So the easiest way to use one of our Go Direct sensors is to literally just plug the cable in. So I plug the cable in, and then plug the cable into your computer or device, and then launch graphical analysis. So this is graphical analysis. I've just launched it. And what you should notice is that because it's plugged in by the USB cable, there's nothing else that you have to do. It's ready to go. I can change some parameters and things like that, but it's already set up. It says force on the y-axis. Let me change this a little bit, make the app preferences here a little bit bigger so we can all see them. But if I just hit collect, I'm ready to go and I'm collecting force data. So it should be that easy. Let me disconnect and I'll just show you how to connect wirelessly. So I'm just going to say select new. Now, if you don't have anything connected by USB cable, it's going to ask you one of three things. It's going to say, hey, do you want to do sensor data collection, which is collect from a sensor? Do you want a data share, which is a way to share data to multiple devices, which is really powerful? Or do you want to use this for manual entry? Like, do you want to kind of use it as Excel? And by the way, this is where that GA Pro code is that you would need to put in. It would show up right here if you needed to actually put it in. So I'm going to connect wirelessly. So I'm just going to select sensor data collection. And then you can see right here in the window, it says, hey, here's all the sensors that I see. And you can see it says GDX hand dynamometer 154. There's also this thing called Proximity Connect. If I bring the camera back up, you should be able to see that that number 154-007Y7, that is the same number that appears right here. So you can see there's the hand dynamometer. So I can either hit connect there, or I could also just take the hand dynamometer. Whoop, there's the hand dynamometer. There it is right there. And I could hold it right next to my computer or over my laptop and tap on Proximity Connect and it would also connect. So that's another great way to connect. So I'm just going to connect. There we go. And I'm connected. Just another thing to let you know about really quick while we're here is some of these sensors are what we call multi-channel sensors, and that's important for our next sensor. Some of these sensors can do more than just what they're mainly there for. Like, so this hand dynamometer can be used to measure force, but it also can be used as a 3-axis accelerometer, and it has a 3-axis gyro. So that's what's another really nice thing is if you get to this dialog, and I'll show you how to get there, you can always get back to that dialog by tapping on this button in the far right corner that looks like a little sensor, and tap on sensor setup and that will come up. So that's what you would do if you actually had it even plugged in over USB and you wanted to select a different sensor channel. So then once again, I'm ready to collect data. Now I'm collecting data wirelessly. See no wires, and you can see that I'm collecting data on the screen. All right. Any questions? That's the basic intro to Go Direct sensors. Graphical analysis is what you want to use. There's some cool features. I can go through that later. But if we're all good, I'll just move on. So if you want me to stop and you have a question, just put it in chat right now and I'll answer it, or I'll move forward. All right. So that was the live demo. I was going to just give you a live demo on how to collect some data, which I just did, just show you graphical analysis and our sensors. But then there's two sensors that we really need to talk about here if we're going to be doing this reflex lab. And one of those is our EKG sensor. So our EKG sensor is really designed to measure EKGs really well. So if you want to record the electrical activity from the heart, you measure across the body axis and you can measure EKGs. But it also is really good for measuring EMGs. EMG stands for electromyogram. That's the electrical activity from a muscle. And then it can also be used to look at rectified EMGs. And what that means is that an EMG is going to have a negative and a positive phase. Rectifying it means I just make all of it positive. So I make all of the spikes positive. Now, the reason why I'm bringing that up is because in the reflex lab, that's exactly what you do, is you turn on the rectified EMG channel. All right, so we don't need to do a live demo of how to set that, or we can do it. I can show you how to get there again. So I'll just connect my EKG sensor. So I'm going to go over here and I'm going to disconnect the hand dynamometer. And I'm going to start over and say new experiment. And then let me bring the video up so you can see this. So there you can see, here's the EKG sensor. There's its number. There's a little light that's flashing. And if I go back to graphical analysis, I can say sensor data collection. And there's the EKG sensor, and I can hit connect. And then remember how I talked about sensor channels? Well, you can change those sensor channels. So the default is EKG, but you can then go through here and select some other ones. In this case, I'm going to wind up selecting EMG rectified, and I'll show you that later, but EMG rectified. There's also this channel called voltage, which doesn't do any filtering at all, but for the reflex lab, you're going to want to select EMG rectified. All right. So reflexes. So the reflex lab that we do uses two sensors. It uses the EKG sensor in EMG rectified mode, and then it uses a force sensor. So this is our Go Direct force sensor, but then you wind up using it with a reflex hammer attachment. So we have this little accessory called a reflex hammer attachment that you can use. And let me bring that up right here. Here's what it looks like. So here's the Go Direct force sensor that you can buy. And then it has a metal sort of casing that you can put on it. And it comes with a handle. And then you remove, this end would normally have a hook. You remove the hook. And then it has two heads. It has this little reflex hammer head for hitting. And then it has this reflex hammer head as well. So two different reflex hammer heads. And then you wind up, hitting things with it. And it'll measure the force. The old lab that we used to do this with used an accelerometer, and it would be problematic because in a lot of cases, students would be asking me, well, what is this peak that I see? What's acceleration? Why am I looking at acceleration? It's going to be much easier to use the reflex hammer to do this lab, and it'll be easier for your students to understand. By the way, you can do this entire lab with our old sensors. So if any of you have our old sensors, like our old EKG sensor, or even an old dual range force sensor, you can still do this activity, and all that you would need to do is get the Reflex Hammer Accessory Kit, and it'll also attach to our older dual range force sensor. So even though I'm showing this with graphical analysis, you could still do it on a LabQuest, etc. All right. Oh wait, I'm going the wrong way. There we go. All right, so I just wanted to remind you once again that This lab is located in one of our two books. We have Human Physiology Experiments Volume 1 and Human Physiology Experiments Volume 2. This lab that looks at simple muscular simple reflexes is in our first book, Human Physiology Experiments, and it is actually called Experiment for Simple Neuromuscular Reflexes. Now, this is a hyperlink right here, so if you get the presentation later, you can just go straight to that activity on our website. The old EKG sensors do not have the EMG rectified option, but don't worry about that. If you just email me at biology@vernier.com, it'll still work fine in most cases. I can even show you how to rectify it. It's not hard. You just need to create a calculated column channel, but that would not be hard. Okay, so have any of you done this lab before? Tell me yes or no in the chat. I just want to know, because what I thought was really interesting, because I used to do this lab every year. I would do this lab every year with my students at Wartburg College, and I couldn't believe how popular it was. There were always students that would tell me every year, or they would tell other students that were coming into my class, like, have you done the reflex activity yet? And then the other one that they would ask is if they had done the goggles activity, because I have another lab that uses, or used to do another lab that would use um, plasticity goggles. But this one was really popular, and I never really understood why until I started talking to students, because I just think of it as, well, it's just a really simple activity. You're looking at a reflex arc. It can't be that complicated. But this reflex is plastic, and that's what they found was really interesting. So it's the plasticity of the reflex arc. It's really simple to do. And then it's also really simple for inquiry. So I would have students every year that would say, I want to do a whole experiment or do this experiment with other people and stuff like that. So it was always very, very popular. All right, so we need to talk a little bit about the procedure. So you need to understand the procedure before you move forward. The key things about the procedure is, if you're going to be doing this lab in class, students have to wear shorts. Someone has to be the subject and they've got to wear shorts. The other thing that you need to know about is you're going to need to mark the location of the hammer strike. There's a spot right along your patellar tendon that's going to be best. So they're going to need to experiment with that. And then you're also going to have to train the students to try to use about the same amount of force with each strike. Luckily, let's see here. I created a video for you. We created a video for you, or for that you could take a look at. You can get access to this. This video is actually in the resource folder, and it shows you how to do all of this. And in this case, I am the subject. My colleague, Sarah Tlarvik, is providing the background. You don't need to hear that right now, but I'm just showing you that's me moving my leg and then showing you where the quad muscle is and then the location of where the electrodes would need to be placed. So then I wind up placing the electrodes in those two locations. You want to place the electrodes across the muscle. So I'm putting one at the tip there and then one pretty far in. And then I'm also placing a ground, and the ground is placed on the side of the leg, the knee. You could also put it on your wrist, but that's where I would place it. So plus and minus, go top and bottom, and then negative there. And then I'm just showing you where the patellar tendon is, right along in there. So right in that location there. And then Sarah is marking it. So that's also what you're going to want to do with your students. And then in this video, we basically go through the entire setup. So if I move forward here, this is just me putting on the electrodes. Where green and red go isn't really that important, but they just need to go across the muscle. Black is the ground, so that's the one that needs to go by the knee. This is all in the written instructions that you have access to, but I just thought we have this video. You can show it to your students. They can take a look at it, etc. There you go. So that's the prep. And then once it's placed, I think my colleague now is going to wind up hitting me with the reflex hammer. Or actually, there's three parts to this experiment. The first part of this experiment is you take the reflex hammer. She's got the reflex hammer there, and she's going to tap it on the table. And then what the subject is supposed to do is every time they hear that noise, they move their leg. So the first part of the experiment is looking at voluntary motor control. So that's the first part. The second part of the activity is the actual reflex arc. And that's where the person will hit the patellar tendon and we'll look at the reflex activity. And then the last part of the activity is also looking at, so here's the patellar tendon reflex. She's going to hit me here with the hammer. There you go. So there's the patellar tendon reflex. Do it a series of times. And then the last part is doing the same activity, this reflex activity, but you wind up doing a specific maneuver that makes it plastic or makes it typically bigger. It's called, I believe it's called the Gendrasic maneuver, and it's just doing this. It's literally just taking your hands and pulling like that. You can also see a change in this reflex by having students read a book or something like that. And that's usually what they find really interesting is all of a sudden this little reflex arc changes. All right, so the key things about getting the lab set up are, like I said, you need to deselect the EKG from the sensor channels. You need to select EMG rectified from the sensor channel list. You also need to select reverse on the meter for force, because if you're hitting, you're going to creating a negative force. So you want the students to see a big giant spike, not a spike going down. It would still work, but it's just better if you do it that way. And then you really just need to change two things. You need to change the rate to 100 samples a second, and then you want to collect data for just 30 seconds. Now, this is also a hyperlink, which shows you how to do all of that. So I wanted to just show you, we've created a little video for you, but that's really what this video just shows is you've got the EKG sensor and the force sensor attached, and then it just shows you going through and changing those parameters. So here I'm going back to connected sensors, and you can see GDX EKG. You open up the sensor channel list, and then you select EMG and EMG rectified, and you wind up deselecting the EKG channel. Or you just want to select EMG rectified in this case. And then this is just me changing the parameters to 100 samples a second instead of 200, and then collecting for 30 seconds. And then once again, just going to the sensor actions right here down below and changing it to reverse and or even zeroing it. So there's the reverse, turning that on. That's pretty much it. And then you're ready to go. All right. The other thing that I just wanted to let you know about that's important is to get the best data possible, you're going to want to use USB cables for this lab. And that's because the reflex is so fast. The lab will still work great if you're using it over Bluetooth, but I would just recommend using it over USB if you can. If you have like iPads or something like that, that's fine. Connect them over Bluetooth. But the latency of the reflex will be a bit longer, and that's just because the Bluetooth takes a little bit of time. So the lab works best when done over USB. So use the USB cables if you can. If you can't, that's okay. It'll still work. The reflex arc may not just seem as fast as maybe you would think it would be, but the lab will still work great. Okay, this is just once again, I've just put up the basic procedure for part one, which is really just, put the reflex hammer together, launch graphical analysis, and then change the setting profile. Okay, so then the other thing that we should talk about is just a little bit about analysis. So in the analysis, what you're going to do is you're going to look at latency, which is going to be, you're going to be looking at the time of the spike from force, and then the time that you see the EMG. That's the latency, or how long it takes for the reflex to occur. The other thing that you wind up looking at in the last activity is amplitude, or the size of the EMG peak, or how tall that EMG peak is. And that's assuming that you applied the same amount of force. Those are the two things that you wind up looking at. Okay, so For these 3 experiments that are in this lab, one is voluntary contraction. So you listen for an auditory cue and then you move your leg. And then there's the patellar tendon reflex where you hit the patellar tendon. And then there's reflex reinforcement of the patellar tendon reflex. What do you expect will happen in each case? Why don't you just tell me really briefly what you think would happen? Go ahead and write that in chat. So for voluntary contraction, what do you think? Is it going to take longer for that to occur, or is it going to be similar to the patellar tendon reflex, and do you know why? Go ahead and put it down there in chat. All right. Anyone? Okay, well, I don't have to go through all of this if you don't want to. Yes, one will take longer because of the signal from the brain, definitely. So just for background, I have these slides for background. They're here if you want them to take a look at. I think they're pretty cool. There's sensory input for the first one for auditory control. The voluntary one takes longer, yes, because the sound must be processed in the brain and then it has to come all the way back down. The simple reflex arc is really, really simple. And then yes, for the plasticity, that reinforcement, you should have the largest amplitude. And these are just really nice resources that are out there if you want them, that I found that are pretty cool. This is the auditory input pathway. This is from the University of Minnesota. It's free on the web that just talks about the auditory pathway and how that information has to travel all the way up to auditory cortex. And then also the voluntary motor control pathway. There's the sensory and motor control pathways, but you can think of it as, for motor control, this is from Oregon State University. You can scroll down here to the motor control pathway. Is that motor control has to come then back down from cortex, through a specialized series of pathways through the spinal cord and then to the skeletal muscles. So you would think that it would take longer. So this is just repeating all of that. This is a multi-synaptic event. Many neurons are involved. Lots of synapses are involved, and it's under conscious control. Okay, for the reflex arc, it should be a lot faster, right? Because it's really just involves 2 neurons, sensory neurons that are sensing stretch in the muscle itself, and then the motor neuron afferents that go to the muscle. So in this case, when you take the patellar tendon, when you take the reflex hammer and hit the patellar tendon, it actually stretches the muscle. That stretch in the muscle itself is then sent through a sensory neuron or sensory afferents into the spinal cord, where they activate motor units or motor neurons that then make the muscle contract. And that's why the leg boosts up a little bit. So it's not under conscious control. It's a reflex. It's monosynaptic. It involves just 2 neurons. So it should be a lot faster. Okay, what about reflex reinforcement? Reflex reinforcement, this is the really interesting part of this lab to me. And the reason why it's so interesting is because the Jendrassic maneuver was found in 1885. But even to this day, I was teaching this in the early 2000s. The mechanism is not well understood why it's plastic. And this little link right here, this is a hyperlink to a recent paper on this that was published in 2019 that basically says, well, we think it's a multi-plastic event. It's a lot more complicated than we thought it was. But the real answer is we still don't know the real mechanism, or I have not been able to find anything that says they know the specific mechanism. But what's really neat is it's plastic, so it changes. And that's a really key thing to talk to students about the brain, is that the brain is plastic, things can change. That's how learning happens, is through plasticity. And so students really like doing that because they're just used to thinking of the brain as a bunch of wires that are all connected to themselves or connected to things. When they're not, they're cells that are connected to things and they can change what they can do. All right, so those are the 3 key experiments. And yes, we would think that voluntary contraction would take the longest, would have the longest latency. The reflex arc would have the shortest latency. And then the reflex reinforcement should have, the EMG should be larger in amplitude. All right, so let's just do a little bit of analysis. Is that okay? Like I want to just show you what the data would look like. So I'm going to open up graphical analysis. I have the sample data. And if you want this sample data, I can get it for you. It's actually supplied. Let me go to the desktop. It's actually supplied in that folder. And it's what we have here is all three activities showed in a data table, but I'm just going to go to the graph. Go to turn on the graph, turn off the data table. And this is normally what students would see is this is activity 1. Yeah, part 1, voluntary contraction. And what you can see right here is the force in blue, right? And then the red over here, this is the muscular activity when it actually happens. So there's a few key things that you probably are going to want to do here to make this a little bit easier on yourself for the students. One is to zoom in on a section, right? So there's like a little section here of 4, and you can hit zoom in, the zoom in button over here, and that'll zoom in for both. It'll keep them together. The other thing that I would recommend doing is going to graph options and turn on interpolate. That's going to make it easier for the students to actually find these points. So if I tap on here, I can go in and I can find the top. Should be able to find the top of that point. There we go. So it's right at around 9.0427 newtons is the peak. And that happened at 9.97 seconds. And then if I go down here, I can find the first height of the peak. And that was at 0.3731. And you would go through and find all of those things to calculate latency. So the difference between when the peak happened and when the second peak occurred, right, that is the latency. Now, GA Pro has a really cool feature here that I would recommend for this activity. And that's because instead of going through and finding out when this peak occurred, because another thing that you can do is you can just highlight a region here, highlight a region, go to view statistics, and it's going to give you the delta Y. It'll give you the max, and it'll give you the time. So that's a faster way to do that. But then you have to go down here and do the same thing and go like, okay, well, and in this case, you would want to look at the peak. So find the highest point, view statistics. And once again, find the max at time 10.25. So that might be an easier way to do it. One of the neat things about GA Pro, though, is you can look at both of these graphs on the same graph simultaneously. So I can go to one graph, and then if I go down here in the bottom left corner, and I go all the way down to edit graph options, I can turn on the right y-axis. And now nothing is shown yet, but I can tap on this other y-axis and I can select, oh, okay, well, I want to look at the EMG. There we go. And in this case, they're right next to each other, right? So it's very easy to see, okay, there's where I could even zoom in on these, but I can then highlight this region, say view statistics, and then it's going to give them both to me at the same time. So blue, that is the force. So the max for force the max force was 5.839 newtons, and it happened at 5.460 seconds. And the max for the EMG, which is in pink here, that max happened at 5.7. So you can see that's about 7.46. You'll be able to calculate the difference there, and it'll make it a lot easier on your students because they can just select each set or each doublet and go to view statistics. So it'll make the lab quite a bit faster. Any questions? So you can sort of see the difference there. I can then also if we go to the patellar tendon reflex, I can turn on force for that, and then turn on the rectified EMG for that. Turn off the EMG there. And you can see that these are all much closer. They're all a lot closer. um, in this case. So let's see here, force. Okay, force, I need to turn off force in that case. There we go. So you can see they're almost right on top of each other. So much, much closer than in the other case. So it's, actually quite obvious. So like if I zoom in on, and you might actually have to zoom in on these. So you can select, highlight it, and then zoom in. And you can see the force here, happens at 11.56 seconds, and the first peak happens at 11.6. So much, much faster, right? So will be much easier to see. And then you can also just compare the amplitude as well for the last activity, part of the activity. So questions? If you don't have any questions, what I just wanted to do was go through a little bit about the data that's there that we've used. You could take a look at. This is in the teacher's notes, the sample data, to just sort of give you an idea. So for like the first 5 kicks for conscious muscle contraction, you can see the average time difference there is about 0.21 seconds between force hitting and actually seeing that response. Table 2, this is actually the reflex, the patellar tendon reflex itself. You can see that on average, it's much faster. It happens, in about, on average, 0.05 seconds, but in some cases, it's much faster. It's faster than that. There's really this little one right here that kind of blew the average off. You can see that it's probably more like around 0.3, between 0.3 and 0.4. So much faster. And then what's interesting is when you look at it without reinforcement and with reinforcement, should be able to see a very big difference. So average height without reinforcement, about 0.3 millivolts, the delta, and almost 3 times as bigger, or at least double the size with reinforcement. So that it actually is plastic. All right, so I see some new messages here in chat. Let me see what those are. All right, so someone asked, how do you know which EMG peak to use when they're so close? Do you mean which EMP, like of the spikes that are there, which one to pick? The thing is to just pick a rule and use it. That's what I say, because the EMG peak will have 3 peaks. If you're using the stats function, it's going to automate it itself because it's going to pick the biggest one. And that's actually going to be easier because then it's just going to pick the largest peak there and it'll use that. But you should be able to see there's going to be a force peak and then an EMG peak right next to it. And you want to make sure you're using the EMG peak that's associated with a force peak that preceded it. Yes, the reinforcement for trial 3, in table 3, that's all the Gendrasik maneuver. That's all just doing this. Really big difference. That's what used to get students really, really excited, is they'd see that and they would kind of freak out and they would go, how is that possible? And I'd say, I don't know. We should talk about plasticity. And then they would design all of their own experiments to figure out like, well, when is it more plastic? How are things more plastic? You can do this reflex activity with students doing math, and it's also going to be plastic. I had students that were athletes, and they would actually ask themselves, well, what happens if I do this reinforcement right after practice? And they found, probably not surprisingly, that it isn't as plastic. But there's all sorts of things that you can do, and you can honestly tell the students, look, we don't know. We don't know what the answer is. So anything that you're investigating is actually interesting because I don't know the answer personally. All right. So I believe that was where I was going to end it there in case you have other questions about anything. Yeah, oh yeah, great. So the ground. The black ground electrode can literally be replaced anywhere. If it's placed on the side of the thigh, that's actually by the bony part of the knee. That's fine. I don't like putting it on the ankle. In our instructions, it says to put it on the ankle. I don't like that because the ankle moves and you don't want to have a movement artifact. So if you place it on the tip of the ankle, then it's going to move. And that's going to cause a lot of noise. Another place that you can put it on that's fine is you can put it on the wrist. So if you just have the student sitting there like on the table, you can also put the ground on the wrist. It's just there to ground the subject. It doesn't actually have to be on the muscle itself. But in the experiment that we did to get all that data, we just put it on the side of the muscle right there on the knee. It worked fine. It's kind of the same thing like if you've done EKGs before. If you're doing EKGs with our equipment, the lab itself tells you to put it on the biceps. It says put the electrodes on the biceps. It's not critical that they're on the bicep. The closer the electrodes are to the heart, the bigger that it's going to be. But if you place one like even on your wrist, left wrist, right wrist, and then on the bony protuberance of your wrist there, you'll get great EKGs that way too. All right. So I think Dylan's just asking you to give us some feedback, see if you have any questions. If you like this, let me know. Once again, all of these videos and everything is available. The sample data is available. So all of these videos that we put together, you can have access to them. You can use in your class. Feel free to give those to your students and use them in your courses. Don't worry about any copyright or anything like that. And thank you so much for coming.